What's up, AP Bio people? So today our topic is cell communication, so let's dive right in. Okay, so first we're going to talk about a super generic overview of cell communication. So our first step is reception, and right now I have the receptor protein and it is within the phospholipid bilayer. And as you can see, we will have the ligand, which is indicated by this fuchsia color. And what's going to happen is it is going to bind to the receptor protein, and that's going to induce this shape change. And so this is a process of reception. Now we go into transduction, where this message, this signal, is kind of changing shape so that the cell can actually understand what is going on. And that's what's signified by these different shape changes. So after this step of transduction, then we go into what is called response, and that is where the signal is actually received and understood by the cell and a response is given. Okay, so now let's go a little bit more in depth. And so our first pathway is called the G protein pathway. And what's going to happen is we're going to have a ligand bind to the G protein coupled receptor, or the GPCR. And so this is going to cause the G protein to shift down this phospholipid bilayer, and phosphorylation is going to occur where the GTP kind of displaces the GDP. And so from there, this G protein is now active, and it once again diffuses along the phospholipid bilayer, and it attaches to the enzyme, causing a structural change within the enzyme, an induced fit. And so after this G protein, it attaches to the enzyme, a response can occur, and the cell can do something with that. A response occurs. And so after this response occurs, then the cycle starts over again with the G protein becoming inactive. And so what happens is GTP is hydrolyzed, and an inorganic phosphate head is released, and GDP is once again what is with the G protein. Okay, so this next pathway we have is called a tyrosine kinase pathway. And so to initiate this, we have our ligands attaching to the structures. And what that is going to cause is that's going to cause a process called dimerization, where the two monomers join together to form a dime. So after that, we have a phosphate group that is attached to the tyrosine branch on each of the structures. And this is catalyzed by the kinase aspect of the tyrosine kinase pathway. And so from that point on, we can have up to 6 relay molecules. So this is one of the most efficient and fastest pathways, because you can have 6 relay molecules attaching to these phosphate tyrosine branches and causing a response within the cell. So for our next process, we're going to be using an ion channel receptor, which is also known as a ligand-gated ion channel. And so what's going to happen is we're going to have our ligand attached to the specific region on the channel, which is going to then open the gate. And from that point, we can have our ions, specifically sodium or calcium ions typically, and those can then flow through the phospholipid bilayer using this channel. And now once these ions have flowed through the cell, and they are now within the cytoplasm, the cytosol, that can elicit a response from the cell, and something else can take place at that point. For this next process, we're going to be using intracellular receptors, which means that the receptors are within the cell instead of within the phospholipid bilayer. And so to initiate this process, we're going to have our hormone or ligand. It's going to simply diffuse through the phospholipid bilayer. And you might be wondering, well, how does it do that? Well, A, it could be hydrophobic, and B, it's small enough to just simply diffuse. And so we have our hormone receptor complex is going to be created, and this is going to go within the nucleus, and it serves as a transcription factor because it allows for the DNA to create messenger RNA that can be used to create a protein. And this transcription of the DNA, this is basically the response that the cell is giving. And so we have new proteins created that will go and do other things within the cell. So now let's talk about secondary messengers, which are non-proteins and water-soluble molecules or ions. So for our first one, let's talk about cyclic AMP, also known as CAMP. So what we start off with is basically a G protein pathway. So we have the first messenger, the ligand, attaching to the G protein linked receptor, and then that's going to cause the G protein to come over and then be activated to go to the enzyme and activate that. The only difference now is that we're going to have ATP being transformed into cyclic AMP camp, and from there the second messenger camp is going to activate this protein kinase A, which is then going to elicit a response within the cell. And so the response could be that the protein kinase A could phosphorylate other proteins and then have a ricochet of responses from that point on. So there are just several things that could happen through this whole process. Continuing on the topic of secondary messengers, we're going to be talking about calcium ions in the form of calmodulin and inositol triphosphate, aka IP3. So we're using another G protein pathway, except instead of with CAMP, our enzyme being adenylyl cyclase, our enzyme is now phospholipase C. And so this phospholipase C is going to break up PIP2 and it's going to create IP3 once it does that. And so now we're going to be using an ion channel receptor, and the ligand for this, or our secondary messenger, is going to be the IP3 that's going to open up the gate and allow for our other secondary messenger, our calcium ions, to flow out of the endoplasmic reticulum and elicit a response. So this concludes our discussion on cell communication. Don't forget to like and subscribe. If you have any questions, please leave them down below in the comments and I'll try to get back to you as soon as possible. Just kidding. Anyway, so everything within this video, this was all original work. All the diagrams were original, the music was original, everything was original. So everything was done by Justice Brown.